This podcast is brought to you by the Albany Public Library main branch and the generosity of listeners like you. What is a podcast? God, Daddy, these people talk as much as you do. Razib Khan's Unsupervised Learning.
Thanks for listening to the ungated version of the Unsupervised Learning Podcast. If you want to read some essays on some of these topics, please check out razib.substack.com. Again, that's razib.substack.com. Thank you.
Hey, everybody. This is Razib with the Unsupervised Learning Podcast, and I'm here today with my friend Alexander Young. And Alex is someone I've had on the podcast before, actually 2.5 years ago, I think with James Lee. You guys can check that podcast on the links. I also actually recorded a podcast with him on the Insight many, many years ago, but unfortunately the Insight archives are gone.
I think there's another one on unsupervised learning as well.
Oh, okay.
The cognitive genomics one.
Yes, so that was yeah. So yeah, it's many times. We're here though to talk about cognitive genomics and everything that's going on with that. We have a new paper. You guys have a new paper that's out, and we will talk about that. Okay, but first, tell us where we are in this field. In terms of what do we understand about heritability? What has GWAS told us? Where are we with family-based studies, et cetera, et cetera. And then, let's talk about the science first, and then we can talk a little bit about like the political controversies, and then we'll go into the Herasight related paper. And actually, I should just have you, speak to your affiliations real quickly. I can tell them, but why don't you tell us?
Sure, yeah. So I'm currently an assistant professor in the human genetics department at UCLA. Before that, I worked, I did my PhD at Oxford, also a postdoc joint between Oxford and deCODE, and I got into family-based studies in mostly because of my time at deCODE, where there was a lot of family data, which was very different from what was available generally in the field. And that's actually proved to be a very important pathway for the future of the field, both in terms of getting a better understanding of heritability, what GWAS is telling us, what polygenic predictions, genetic predictions of traits are telling us, and also in embryo selection, IVF, which I've been working in with a company, Herasight. So I don't know if you want to, should I go into some detail about GWAS or what should I talk about? That's a broad topic.
Yeah. So GWAS is a buzzword that I think a lot of the listeners have heard. Like, tell us what a GWAS is, and tell us how it's useful in understanding heritability.
Yeah. So GWAS stands for Genome-Wide Association Study, and this became the key study design in human genetics since about 2007 to 2010, and it's really dominated the field since then. And it's a very simple study design. All that you're doing really is looking for correlations between genetic variants and diseases or traits like height. And it's actually simpler than the studies that went before, these linkage studies that were based on pedigrees and families, which were kind of cool. But the GWAS design, which is much more just a simple regression of phenotype onto genotype, that ended up being much more powerful and simpler, easier to gather large amounts of data in these big meta-analyses as well. And that's kind of revolutionized the field. There were a lot of critics of GWAS early on, because especially people that thought it wasn't going to tell us much about biology, and they were partly right, partly wrong, I would say. So the original impetus for GWAS was to find genetic variants that were strongly associated with some like disease, like diabetes. And we found some of those early on, variants associated with like body mass index, diabetes, height, whatever. And from those strongly associated variants in the population, we would be able to figure out something about the trait's biology. And that was a moderately successful research program. But yeah.
Well, actually, let me just interrupt real quick. So, when you talk about traits biology, just to give the listeners an example. So you do GWAS on height, and there's something called Gene Ontology Go, the categories of like, what are the genes doing functionally, it turns out that a lot of the height-related genes are involved in bone morphogenesis. Okay, this is, as you know, and this is exactly what you would expect if the GWAS is actually zooming in on real biological pathways of relevance to the characteristic, right? And you see something similar with, the cognitive genomic stuff that you're interested in, correct?
Yeah, although the cognitive genomic stuff is a bit tricky. I mean, I think these brain-related or cognitive or educational traits tend to be what we call highly polygenic. So they're affected by many, many genes across the genome. And there's not really any one particular gene that really stands out strongly from these GWAS studies. Now, sort of skipping ahead, I guess, to a different design where they look at rare variants that have a damaging effect on a protein that sometimes they cause the protein to completely lose its function. They've more directly identified genes associated with cognitive ability than the typical GWAS studies. So in terms of discovering biology, GWAS has been moderately successful, and it varies from trait to trait and disease to disease. And now we also have these related study designs that look at rare variants that have a known functional impact on particular proteins, and we can use that to sort of directly identify, if this gene is broken, then it messes up this trait or this disease or something. And pharmaceutical companies are very interested in that because you can identify a drug target from that. So that's another kind of study design that's sort of evolved out of this GWAS paradigm.
Yeah, and so there was this whole issue. So GWAS, you're talking about GWAS. Relate to what heritability is and how heritability is different, because I think the two are confounded or conflated in a lot of people's minds.
Yeah, people do confuse this. I was helping Scott Alexander with a post he was doing about missing heritability recently, and I kind of realized that this, even for smart people who are outsiders in this field, it kind of gets confusing because there are all these different study designs and different ways of measuring heritability and how much of a trait we can predict, and they're kind of related to each other, but they're not exactly the same. So it can get a bit confusing for outsiders to understand. And so with a GWAS, we're not really trying to estimate heritability with a GWAS or with the GWAS data. And typically we only have data on what are called common variants. So these are variants that the minor version of the variant, so you have an A or a T, the less common one is present at a frequency of, say, at least 1% or at least 0.1%, which is actually only a minority of the variants in the genome. But from that GWAS data, you can estimate something people call SNP heritability, and that's the amount of trait variation you can explain with a linear model of the genotypes that you have available for your GWAS. And that can put an upper bound on how much of the trait we can predict using that kind of data that we have for GWAS with what we call a polygenic score. So a polygenic score takes the GWAS results across the genome, and it does, some of these methods are quite sophisticated, but at a basic level, all it's doing is taking the associations from a GWAS and using them to add up the estimated effects of all these different variants across the genome in a simple kind of linear model. And that is another measure of explained variation. So you could have, say, for something like height, the SNP heritability might be 45% when we estimate it from GWAS data. Then we have a polygenic score, and they're actually very good for height now because we have sample sizes around 5 million that are available for height. So the polygenic score might mean we're able to actually predict around 40% of the variation in height. And then this is in contrast to more traditional.
Oh, let me just be clear, reiterate, this is population-wide variation.
Yes, this is the variation in the population you're explaining, and actually it's a little complicated because it's not necessarily the GWAS type estimates of heritability and what we can explain with a polygenic score. They're not necessarily just capturing the causal effect of genetics. They can also be capturing gene-environment correlations or confounding, or it gets a bit complicated, but like assortative mating can also inflate these estimates. So they're not as well controlled estimates of heritability as the classical estimates of heritability that mostly come from twin studies. So twin studies, the classic twin study design compares the correlations between identical monozygotic twins and non-identical dizygotic twins. And the key assumption there is that any greater correlation between identical twins and non-identical twins is due to genetics, because they both are raised in the same kind of family environment. And it seems like a reasonable assumption that, and for example, with height, those estimates usually come out at like 80%. So this is a bigger number than we get from the GWAS data, which is say like 45%. Now there are various complexities in comparing those numbers, but this gap has been labeled the missing heritability problem, and that's something we could maybe go into a bit more detail on if you want.
Yeah, so missing heritability just means that you have these twin studies that show up. I think it's like you could say beyond twin studies, just like classical design of these sorts of inference of heritabilities, because it's not just like in human constructs. Like heritabilities obviously exist in animals and stuff, and agricultural genetics. And then what happened initially, I think, with missing heritability, and I'm talking about like say 2010, around that period, is you had these genotyping arrays, with hundreds of thousands. Actually, like at that point, like sometimes it was like tens of thousands of markers, and they were not picking up the heritability and the hits on the GWAS, right? And so statistically, you knew that it was supposedly there, but you weren't figuring it. You weren't pinning down the causal variance in terms of like a molecular genetic, mechanistic fashion, right?
Mm. Yeah. There are different sort of notions of missing heritability. One is like, I think I've heard Eric Turkheimer kind of propose this, that we should only really consider heritability explained if we have like an exact causal mechanism for each variant. We know the exact causal variant, we know its exact biological mechanism. I would say that is not a widely accepted definition. And then there are more sort of statistical notions of like how much of variation we can explain, even if we don't necessarily know which causal variants are explaining it or through what biological mechanisms they're acting. And that's sort of more where the debate is now. But yeah, this debate is sort of, it does relate to how well we can predict traits genetically, but they're not exactly the same thing. And I've seen a lot of people get confused. Some people intentionally, I think, sow confusion about this. But like if we can only predict, say, 2% of the variation in a trait, but we actually know that from the GWAS data, if we had a bigger sample or something, maybe that could go up to like 20%. And it doesn't mean that there's only 2% is the heritability, and you just have to be careful about these different notions, like what we can predict versus what we could in theory explain, and then the maximum possible that we might be able to explain if we could account for all genetic variants, not just the ones that we have for typical GWAS data.
Yeah, at this point, like, when it comes to missing heritability and these sorts of things and understanding causality, you're getting into, deeper philosophical waters, right? But I would say that in terms of explanatory power, for the regular person, if you can get a medical test and that gives you a certain polygenic risk score from certain variants, I think for them, that's causality.
Yeah, the polygenic score performance is a bit more concrete and directly relevant to real world applications. Like there are promising results now looking at how we can use polygenic risk scores for prostate cancer or breast cancer to improve our decisions about when to bring people in for like an MRI or some sort of other screening test. So we can measure the performance of these genetic predictors quite concretely and reason about whether they're going to be worthwhile for various applications. The missing heritability debate is more abstract and maybe academic. I think it's important because it does get at this more fundamental question of like nature versus nurture. To what degree, especially when we talk about things like socioeconomic outcomes, intelligence, education, then this missing heritability debate, like it does get to fundamental questions about inequality in society and things like that. And then the relevant parameters are maybe less how well other, we'll talk more about the applications of genetic prediction of intelligence, but at a more high level, societal level, then knowing to what degree are the differences in school outcomes or intelligence, are they genetic, then that is important. And there's still an ongoing debate, like, are twin studies overestimating heritability for these traits? I think there is evidence, actually, that they are overestimating heritability for educational attainment. We've seen that by comparing. So there are more robust methods for estimating heritability. One uses relatedness variation between siblings, which is basically random. It's due to random segregation of genetic material during meiosis, like whether you inherit one or the other copy of each allele from your mother and your father. So this varies between siblings. So siblings can vary between like 60% related and 40% related, roughly. And that random variation can be used to estimate heritability. An interesting study done by Loic Yengo and Peter Vischer in collaboration with 23andMe, this preprint just came out a month or two ago, so this is very cutting-edge stuff. They put together a sample of about 500,000 sibling pairs, meta-analyzing 23andMe with a study that I did in deCODE and then some other studies that they did. And they found, I think these provide actually probably the gold standard heritability estimates now. They're sort of eclipsing twin studies. And they put the heritability of height at like 70%, BMI at 50%, cardiometabolic disease, I think, was around 26% or something like that. So I think, well, then you have to adjust. There's an assortative mating thing as well. So actually, these methods and twin studies can also underestimate heritability due to assortative mating. So if you account for that, then I think their estimate for the heritability of height was something like 86%, which is, not that far off what twin studies are doing, but you have to be careful about the comparison with the assortative mating thing. So it looks like maybe twin estimates are still a bit higher than we're getting from these more robust genomic methods. But unfortunately, we don't have really precise estimates from these more robust methods. The sibling-based method, there's also a method I developed called relatedness disequilibrium regression, which is like a generalization of the SIB version. I know some people now working on applying that to things like school grades and cognitive outcomes in kids. So we're hopefully going to get some estimates from that in the near future. But 23andMe actually, we used to in the SSGAC, used to collaborate with them on doing education GWAS. We did a 3 million person GWAS of educational attainment. Most of that data came from 23andMe, but they had a change of heart at some point around 2020, 2021. I wonder why. And then decided that it was unethical or bad publicity or something to do any GWAS of educational attainment, and actually they wouldn't allow I don't think the Vischer and Yengo group to study the heritability of education or other cognitive and educational outcomes 23andMe have, which is a shame because there's so much debate about that online, and they have some good data on it, and they're not letting us look at it, unfortunately.
Yeah, I mean, so I do want to talk a little bit about the political aspect, but let's like dig a little bit more into the science. Can you explain to me why? I mean, I think I know because, well, I mean, whatever. Like, I'm in a different state of knowledge about this sort of stuff than the listener. And so I am following what you're saying, but I just started realizing when you're talking about disequilibrium regression and, what I'm trying to say. Yeah. So when you say assortative mating distorts heritability estimates, tell us why, Professor.
Right. So assortative mating is when parents are more alike than random pairs for certain traits. And this is actually pretty strong for education, intelligence. Height is an obvious one. You can see that people, if a woman is very tall, she's often with an even taller guy. And this, over many generations, has the effect of increasing the genetic variation in the population because it induces positive correlations between alleles that affect height in the father and alleles that affect height in the mother. And this inflates the variation, the genetic variation in height over many generations. So this is kind of a complicated thing to think about, but basically in the twin estimates and the siblings, so all the family-based estimates, they basically don't account for the inflation of genetic variance due to assortative mating, so they're downward biased because of that. And there are ways to adjust for that, but it's actually it's not trivial to do it in a way that is completely without assumptions or whatever. So there's always certain assumptions one has to make. But basically, yes, this applies to estimates of heritability for intelligence and education and height, that the ones coming out of twin studies and sibling-based ones, or this RDR method that I developed, they're all downward biased by assortative mating. But then other estimates from other methods can be upward biased by it. So, the fact that humans don't mate randomly just it makes the lives of statistical geneticists much more complicated, although maybe more interesting as well, I would argue.
And so, actually, I had Greg Clark on the podcast to talk about his work on heritability and social status, and you kind of got involved in that discussion.
Yeah.
Can we relitigate that a little bit in terms of like what your take was on that?
Sure. So Greg Clark put together this very impressive data set looking at, I think he had various outcomes like wealth, literacy, some sort of general measure of socioeconomic status. And he looked at how that's transmitted over generations. This was using sort of marriage and other kinds of records he put together going back to like the 1600s in England. So it was a cool data set. And I mean, this actually goes back to one of the foundational questions at the founding of quantitative genetics, which is correlations between relatives. So Sir Francis Galton, cousin of Charles Darwin, he coined the term regression in relation to this phenomenon he observed called regression to the mean, or mediocrity, I think he maybe called it in the original parlance, which was observed for height in the offspring. If you look at the average height of the parents, then the height of the offspring is correlated with that, but it tends to be like a little bit lower. So the prediction is a little bit lower than the average height of the parents. And then Fisher, in his R.A. Fisher, so he's kind of the founder of quantitative genetics and one of the most important figures in both the foundations of statistics and population and quantitative genetics. And he wrote this paper in 1918, which really it just has, I don't know if it's as equivalent in many other fields where there's one, maybe Newton in physics or something. It's almost like Newton in physics. He kind of like wrote this paper that basically set the foundations for so much of what we still do today. And one of these foundational questions was, how can we explain the correlations that we see between relatives? So parents and offspring are correlated. Parents and then the offspring, then the offspring's offspring, their grandchildren are also correlated. So you can compute correlations between all different kinds of relatives, including in-laws. And you can think about what kind of models can explain those correlations. So that's what Greg Clark did. He took this model from R.A. Fisher in his 1918 paper, which looks at how you can explain correlations between relatives based on genetic inheritance and assortative mating. Assortative mating tends to increase correlations between relatives, so especially more distant relatives. So that generally, without assortative mating, under a linear model, you expect that the correlations between relatives decay linearly. So siblings would be half as correlated as monozygotic twins, and then cousins would be 1/8 as correlated as monozygotic twins, and this just all linearly decays. When you have assortative mating, it doesn't decay quite linearly. You have increased correlation between more distant relatives. So from this pattern of correlations between different relative classes, you can fit a model based on Fisher's 1918 paper, which is what Greg Clark did, to try and understand, can we actually explain all of these correlations just with genetic inheritance and assortative mating? And Clark's paper made quite a remarkable observation that just those two parameters, if you fit them, can actually explain these correlations pretty well. However, just because you can fit a model with a simple model that explains a set of data, it doesn't actually mean that that model is true. And I think that some more there's been actually some pretty interesting papers coming out of the Scandinavian countries where they have these national register data sets where they can look at huge numbers of relative pairs, including in-laws. And they see that maybe the simple model that fit Greg Clark's data doesn't quite fit the more complicated set of relatives they can look at in these national registers. And I think there is some evidence, especially for even in Greg Clark's work, I should say, there's evidence that there's non-genetic transmission of wealth, which makes sense. I mean, we know there's a mechanism for that. People leave wealth to their offspring in their wills, so it clearly can't be purely genetically inherited wealth, right? So we see that in the Greg Clark data, and then even for some of these more educational outcomes, we see stuff that doesn't quite fit that simple Fisherian model that just has additive genetic inheritance and assortative mating. There's probably some cultural transmission from parents to offspring as well.
Yeah, which is, I mean, cultural transmission from parents to offspring, though, that's like, I would say like that is the Western null, though, right? And so I think that that is the radical aspect of Greg Clark's model and this way of thinking where heritability in a genetic system, so we're talking about heritability, I mean, like the colloquial way to think about heritability is, well, it's just like parent to offspring. But when we're talking about it, just for the listener to be clear, heritability is, you know, the proportion of the variation of the population of a trait that can be explained by genetics, specifically genetic variation, you know? And so I think the interesting aspect of Greg's thesis is that over the long term, and, you know, this thesis has been worked out in like many books, or not many, but like, you know, multiple books now, is that there is something to social status, which it's, you know, that's a downstream variable, right? But there's something where social status is like a bundle of like cognitive characteristics, perhaps behavioral. Yeah, exactly. Yeah. And like, you know, and that is
health as well.
Yeah, and that's like quite stable. And there's some like, you know, weird results from the economic historical literature of like, you know, the most prominent like financial families, like, you know, in finance in Italy in like the 19th and 20th centuries, their roots go back to the 14th century. That's like really crazy if you think about how many centuries that is, right?
All these sort of Norman surnames that are still overrepresented in Oxford and Cambridge and England and in military generals or whatever.
Yeah, so I mean, I think the Norman data, just to you know, recapitulate, is basically, you know, you have like about what actually the proportion of surnames have increased apparently from like, you know, less than 0.5% to like about almost 1%. So there's been some elite, you know, over reproduction of Normans probably.
Or people preferentially taking
if they hit the elite status, yeah. So, but these Norman surnames are like, you know, in some fields are still like I think like 10x overrepresented. You know, 1,000 years later, I think that's what you're. I mean, that's like a massive like retention of elite status.
Yeah, the Clark has definitely documented this remarkable sort of persistence over generations, and it fits very well this simple genetic inheritance plus assortative mating model. Now, I argue that I don't think that model I don't think it's a true causal model, and this is why we need the genomic data ultimately. Like this is the advantage of having real genomic data on families, as well as the Greg Clark kind of data is great, but it doesn't give us what we'd call causal identification. So you're just looking at these correlations and you try and explain them in different models, but we can't necessarily identify, is it really genetic causation or is it cultural transmission? I mean, I think he makes a strong his data makes a strong argument that clearly genetic transmission is playing an important role. But this is why I think we still need these very robust genomic database methods using families where we have this randomization. It's like an experimental design that enables us to really identify the causal component of heritability in the current generation. Now, things can change over time as well. That's another factor. But I think that, you know, the combination of those two kinds of data, this sort of long historical or national register data with genomic data that allows us to do causal identification, that's going to, I think in the near term, we're going to have a very clear picture of the causes of, you know, correlations between relatives and persistence of inequality over generations.
Well, I guess the question that I have, when you say near term, like what does that mean? Like, I mean, like 10 years, 50 years?
I think less than that. I would say 5 years. Well, again, it depends a bit on what we're allowed to do. A lot of these data are probably out there, but are we
all right. You know what? Like this, we can like pivot a little bit. I do want to like touch back on the science, and we're going to talk about the Herasight paper.
I'm not trying to bring it all onto
No,
politics.
no, but like
that is relevant to that question.
Let's pivot. So the Young paper that you were talking about, they were not allowed to look at certain things. Like they specifically said they were not allowed to look at certain things.
Yeah, they weren't allowed to look at educational outcomes.
Basically. Well, why? Education's good. Should we want to understand that?
Well, I understand people are sensitive about looking at education, cognitive outcomes, but I think especially when you're just looking at the heritability measure, I think it's a little bit overblown that for them to be so sensitive about it. I mean, I can understand them maybe being sensitive about releasing all the like GWAS data that would enable you to construct a polygenic predictor for education, from which they never did. Even when they were collaborating with SSGAC, we were never able to release all of the data that we had for that. And that's also, their business interests, which is fair enough. But yeah, I think for estimating heritability, I think they should let us do that. It doesn't seem that sensitive to me, and everyone's arguing about it anyway, so they may as well just help settle the argument, in my opinion.
Yeah, I mean, but some people don't want the argument settled, right? I mean, I guess, would you say, I mean, you live in you live in the United States, you're an American institution, you've been here for a while since your graduate work, level work. So, I mean, do you think this is a particular American sensitivity, or I get the sense in your field, there's a lot of work going on in England, but also in places like the Netherlands, and they don't seem as sensitive as Americans. And do you think that this is particularly just like an outcome of the racial issue, or is there other things going
on? I think America has been particularly sensitive. So the NIH in particular has kind of blocked a lot of this research. Actually, James Lee, who we were on a podcast here together before, he wrote an editorial in City Journal detailing, and I know a lot of people have experienced this firsthand, and most people are afraid to say anything about it, basically. But I'd hope now with the new administration that they'll open this up. But basically, not in every NIH data set, but in a lot of NIH data sets, you weren't allowed to study the genetics of education or intelligence because it was claimed to somehow be stigmatizing. It was never specified whom was being stigmatized. I don't really understand that reasoning myself. It doesn't make sense. I think it was sort of ideological blocking of research in publicly funded data, which I think is illegitimate, really. I don't think that there should be that kind of sort of hypothesis-related blocking of research in NIH data, and I hope the new administration will change that. And it didn't even, actually, they extended it. It was also applied to some like alcohol and like BMI phenotypes in some data sets. They said it was too stigmatizing, to almost trying to cancel in certain data sets, really cancel the study of the genetics of a lot of different traits, which deserves to get more attention. That it's still sort of ongoing as well. I don't think it's really been changed yet.
So obviously, there was a lot of like political stuff in your field, in and around your field. Okay, like the whole, I remember seeing stuff. I'm not gonna say who, just friends of mine who are in, study do GWAS in health and stuff like that, and they basically were saying on social media, these social genomics people, they are making our work more difficult, and we're trying to study serious things, as in like disease, like the risk for type 2 diabetes or the risk for cardiovascular disease. And, I think the attitude, I mean, actually, I know this is the attitude because people have told me this privately. They're just like, social genomics is, it's just not that important. And so why is our field taking heat for this sort of stuff?
Well, I'm not sure that anyone really believes that it isn't important. I think that they just don't want the controversy of that field to be associated with what they're doing, their sort of biomedical research, which I understand from the selfish perspective is maybe makes sense to avoid any of this controversy and just say, oh, all we're doing is like better polygenic scores for diabetes and cancer, which is important. I'm very much in favor of that research, but I think we have to ask ourselves the question, like, are we in favor of free and open inquiry or not, as a society? And if we are, then we should be allowed to study the genetics of intelligence and the genetics of group differences and things like that. And I know a lot of people are opposed to that, but ultimately, if we want to have a full and clear understanding of human genetics, these things are not really independent either. Like, the genetic effects on intelligence and education also affect health, right? We see that in some of the research Herasight's been doing, that the genetic predictors for IQ also predict lower cardiometabolic disease risk. And you've been blocked from studying the genetics of IQ in data sets that were explicitly set up and funded by taxpayers to study the genetics. Well, not just the genetics, but to study the underlying causes of cardiovascular disease. One of those underlying causes is related to the genetics of intelligence, and they're saying you're not allowed to study it. Ultimately, science can't be walled off into oh, this is biomedical and this is all good and nice and moral. And then there's this social genomics stuff that is bad. I don't think that's actually how science works. And you can't just have censorship in one domain of the field and it not affect others. That's an unrealistic model.
Well, I mean, so yeah, okay. Broadly, I think anyone who's listened to me for a while knows that I totally agree with that. There's no gap between our views. But I do have to say, science has social consequences, right? And people are worried that if you do this sort of study, basically, here's a thought experiment. I don't want to. Look, you've already been in the hot seat, so I think I could ask you these sorts of questions. You know what I'm saying? I wouldn't normally ask you, but it's what if it turns out that your research results in work that shows that different groups have different cognitive profiles, right? What would you say to someone? Because you're just interested as a researcher in the genetic architecture of traits and characteristics, and potentially their evolutionary history, whatever. I know how your kind of people think, but there's people out there in the world that find that they read that Ashkenazi Jews have higher IQs because of their cognitive profile, because of their genes, right? And how are people going to react to that?
Well, I think that on that debate specifically, it annoys me that everyone seems to think they know the answer to these questions before we've really done the studies. And I don't take a strong prior position on that. And I think that these are important questions for society. And I'm not sure pretending that we know the answer when we don't, or lying to ourselves about what the answer might be because we find it politically or morally convenient. I don't think, one, it's not sustainable because these studies are going to happen one way or the other, whether people like it or not. We're going to be entering a world of data abundance in the near future, not just in the US and Europe, but also in China and East Asia. And these studies are going to get done whether people like it or not. So do we want them to be done by, professionals who know what they're doing, or do we want them to be done by random anons on the internet? And I also think that the people who want to stop these studies, they seem to have this weird contradictory thing where they're oh, there's nothing to see there, but don't look.
Okay, that's a fair point.
And I don't know if did you see the study from, I'm a co-author on this with Peter Vischer and
the Mexico City one? Yeah, talk about that.
Yeah, so in collaboration with the Mexico City Perspective Study, which is this really it's quite amazing now, actually, I'd say. Mexico now has a world-class biobank, the Mexico City Perspective Study. It's competitive, I would say, in the quality of the data with what we have in UK Biobank. It's better than a lot of the US biobanks, to be honest. And what's interesting about this Mexico City data set is that they have a lot of family data. So that enables us to do a study design that people have thought about before to look at the causes of group differences, called what we call a within family admixture study. So the samples in Mexico City are primarily admixed between European ancestry and indigenous American ancestry. And there's also contributions from African and East Asian ancestry, but they're much smaller. And there's quite a lot of variation in the proportion of the genomes that the samples get from European and from indigenous American sources. But if we just correlate the genome-wide ancestry proportions, say, like, comparing people that are 50% European to like 70% European, and looking at how much education they have or their height or their type 2 diabetes risk, then this is confounded with environmental effects. So, I've been to Mexico City once. I think it's a great city. But I'm told by people who know better than me that it is a very stratified city, actually. It's quite socially stratified between the different neighborhoods, and that social stratification correlates with ancestry. So simply comparing ancestry proportions between individuals in the population is confounded with things like which neighborhood you grew up in, or the wealth of your family. Maybe the more European families are from have some of this Greg Clark thing. They have this persistent wealth. It might be discrimination based on ancestry. All these kind of things going on, right? So one thing that you're able to do in the Mexico City perspective study is look at the correlation between ancestry proportions within a family. So siblings vary. Say one sibling might be like 55% European, the other one might be 50% European. These variations are pretty small, but the Mexico City perspective study is large enough that we have power to actually see within family what is the effect of variation in European versus indigenous ancestry components. And interestingly enough, we saw for height and type 2 diabetes that there's a strong correlation between indigenous ancestry and having lower height and a higher risk of type 2 diabetes. The question is that genetic causation, or is it some sort of like confounding? Now, it appears for those two traits that it's basically entirely genetically caused, I would argue. I mean, there is this complexity of this. So there's this famous thought experiment called from Yanks, the red hair gene experiment, where if someone inherits the gene for red hair, then maybe they get hit on the head by the teacher or somehow.
And they have no soul.
Yeah, according to South Park, at least. Or, discriminated against in other ways, and that leads to these outcomes. So even with this study design, we can't confidently say what the mechanism is. We can say there's a causal effect of ancestry differences, but it could be mediated through social mechanisms, including ancestry-related appearance discrimination. Personally, I doubt that's what's going on with height and type 2 diabetes. We see that basically it's the differences by ancestry seem to be pretty much entirely genetically caused. And I think that's important to know for biomedical science, actually. The type 2 diabetes thing is a really important thing to understand, that, part of what is causing variation in type 2 diabetes risk in Mexico is variation in European versus indigenous ancestry. And ignoring that, it's not going to help us understand who's at risk of type 2 diabetes and why they're at risk of it, what the causes are, right? And you can make the same argument for why some people get more or less education. But now it turns out we did this analysis for educational attainment as well. And while there's a strong correlation between having more indigenous ancestry and lower education level in Mexico City, within family, the effect is basically zero. So this goes against the sort of what we call hereditarian way of thinking, which would have predicted that there would be that difference would be explained by genetics. And basically, when we look at the within family effect, it's zero. So it seems like in Mexico City, genetic differences between European and indigenous ancestry don't actually explain this. And it is some sort of, stratification going on, some sort of confounding. So I think that shows that, people that are really afraid of this kind of study design are misguided, because by doing this study, we've actually done more to disprove some of the stronger forms of hereditarianism than all the people that shout about how terrible it is to study this have ever done.
Yeah, I saw that result, and I was like, it's interesting. It's like science says you be woke now.
Right, yeah. I mean, it was a sort of politically correct result, but we don't. That's why we need to do the studies. We actually don't know. We don't know what the answers will be until we do the studies.
Can I just say it do be like that, Mr. Sansel? All right. So we've been talking for a while now. I guess at this point we should probably talk about Herasight. It's a company that you have been doing some work with, and it kind of caused some controversy, I guess, in academia when it came out that you were working with them. So talk a little bit about that and why you are working with Herasight, and what you're doing for them.
Sure. So Herasight is a company developing advanced genetic tests for IVF. So when people are undergoing IVF, they produce a certain number of fertilized embryos. And then, there are existing what are called pre-implantation genetic tests that can be done on these embryos. So one of them that's pretty well established is called PGTA. So this is a test that looks for aneuploidies, so chromosomal abnormalities such as Down syndrome. It's not too controversial, though I did discover recently it's actually banned in certain European countries. Then there are other kinds of tests that are done. One is called PGTM, which looks for Mendelian disease genes. So if a couple say they both carry like cystic fibrosis genes, then there's a 1 in 4 chance that their offspring is going to inherit one copy from each parent and then have cystic fibrosis. So you can sort of screen the embryos for that. So the idea of genetic testing in IVF is sort of quite well established, but there's been a sort of promise of using what are called polygenic scores, which I was talking about earlier. These are these genetic predictors of disease risks and traits that are derived from GWAS data, and these use the whole genome, basically. These use the whole embryo genome to create a prediction for, say, what chance you have of getting type 2 diabetes or breast cancer, or what your IQ might be. And for some reason, this idea is incredibly controversial in academic human genetics. And it's one of those odd cases, and I think there's a lot of this in academia, and obviously I know the genetics case well, but where there's sort of a narrow orthodoxy about this technology that everyone knows that you're not allowed to disagree with. If you disagree with it, you're going to suffer career consequences. So there hasn't been really anyone particularly prominent or credible involved in this industry, I guess, until I came out as being involved with Herasight. And I think this has actually hampered the development of the technology. So if there's sort of taboo about it, if the purpose of it was to hamper the development of the technology, then it's sort of been successful because it's put off. Although I know a lot of academics in private tell me that they're in favor of the technology or at least neutral about it, in public, they won't say anything about it because everyone's afraid of the career consequences or whatever, which are real. And I've already suffered them out. Actually, I've already had one job offer kind of rescinded, basically, after this came out that I was involved in Herasight. And I've had some collaborators pull out of papers that I had in review at like Nature. So for whatever reason, and I think that I can speculate on those reasons, although I don't know how useful that is, there's a taboo about this technology in academia, even though really it's quite clear that it works and it has potentially huge benefits. Like, so Herasight launched on social media just back in, I think that was August, and they'd been operating in stealth mode for a while, in part because we know this is controversial and we wanted to build up our research and make sure we had everything properly validated before we came out. Unlike some other companies like Nucleus in this space that have acted pretty irresponsibly, in my opinion. There's a couple of other companies also operating in this space that have been around a bit longer. And we came out with, the most powerful genetic predictors for various diseases like type 2 diabetes. For example, with our predictor for type 2 diabetes, you could pretty much half the risk of getting type 2 diabetes if you selected among a decent number of embryos, like 5, 10, 20 embryos. And for parents with a family history of type 2 diabetes or affected themselves with type 2 diabetes, this can be quite a dramatic risk reduction in terms of absolute terms as well. Like, I think maybe it can go from a sort of 40% chance. If you have two parents that are both affected with type 2 diabetes, you can, say, reduce the risk that the offspring has type 2 diabetes from 40% to like 20%. More controversially, Herasight have also developed by far the most powerful genetic predictor of IQ. And they actually, this is Tobias Wolfram has masterminded this. I haven't been involved actually in any of the
That's an
prediction
interesting
stuff.
word to use, that he's masterminded it. It sounds like you're making him sound a bit like a villain.
Well, it's been a long thought out and strategized plan by Tobias in how to do this, and he's developed some pretty innovative methods based on like phenotype engine, like figuring out how to squeeze the most information out of all these data sets like UK Biobank that are out there to maximize your prediction. And it's partly a matter of incentives that academic organizations haven't had the incentive to create the best possible predictor of IQ, in part because doing that doesn't necessarily help your career because a lot of people don't want you to do that. But so the Herasight IQ predictor has a correlation with latent general cognitive ability, which we call G, of just over 0.5. So what that means is if your polygenic score for IQ, the Herasight one, was 2 standard deviations above the mean. So that would be sort of close to an outlier score. That would imply that your general cognitive ability was like 1 standard deviation above the mean. In IQ terms, that would be like 115 points if you were comparing to a mean of 100. So this is much more powerful than people thought would be possible based on GWAS data. And translated practically into embryo selection terms, it means that you could get a boost of up to 9 IQ points. So this is if you have a European ancestry couple selecting among 20 embryos, you'd expect a gain on average to be around 9 IQ points, which is pretty large, really.
That's 2/3 of a standard deviation.
Yeah, and I should point out that's on the latent general cognitive ability scale, not on the measured IQ scale. So the effect on the measured IQ scale would be a little bit smaller because measured IQ is kind of a noisy measure of the latent ability scale. And Tobias has done a lot of interesting, psychometric analyses in that paper to show that this makes sense, this result, that the effect is larger on the latent scale because if you think about it in the embryo selection context, we're affecting the genes of someone at conception, and that's going to affect their latent trait, not some noisy measure of it, which is sort of downstream of the latent trait. But yeah, this isn't actually why I got involved in all of this. I can tell my story of why I got involved in it is I remember I was at a dinner with various professors, and we went around and talked about what we thought the most important application of all of this GWAS polygenic score stuff we were doing was. And I actually found everyone's answers kind of lame. Like they weren't that exciting to me. And it seemed kind of obvious to me that this embryo selection application was maybe the most important and the most exciting, but people were afraid of it because of the taboo about it. But obviously, it can be very effective. Like anyone that really understands the genetics of this would understand that this can work. And the people telling you it can't work are either stupid or lying to you. And I think more often they're lying to you, to be honest. And I was interested in getting involved in this field, but it looked like a lot of the companies, other than Herasight, weren't really serious about doing R&D, like innovative R&D. And so Herasight actually approached me with this idea, which I found quite intriguing. So I think I mentioned before, there's this already established test. It's used in 60% of IVF cycles in the US currently, called PGTA, which is this test for aneuploidy, such as Down syndrome. And what this test does is that they take a biopsy from an embryo of a few cells, and they generate ultra-sparse sequence data on this. So what that means is you're getting information on, say, 2 to 6 in 1,000 sites in the genome, which is very, very sparse compared to the information people normally get to, get a genome on someone. And they approached me with this idea, like, can we develop an algorithm and an approach that enables us to use this routine data used in 60% of IVF cycles in the US? There's loads of data already out there. Millions of embryos already out there have this data. and it's used worldwide as well, not just the US. And I was pretty intrigued by this problem, both from a scientific and technical point of view, but also for the practical implications, because currently there are these companies, Orchid and Genomic Prediction, and they are where you need to go to get the data, genome data, whole genome data on an embryo that enables you to do this prediction of traits and disease risks. But this idea was, can we take this routine data and do the same thing? And we developed an approach that enables us to do that by combining it with genome information on the parents in this innovative sort of machine learning algorithm based on hidden Markov models that we designed to infer we can basically reconstruct the whole embryo genome now just from this routine data combined with the with data on the parents. So now we can take this to like most IVF clinics in the US and many around the world. People can just go to the IVF clinic, and in the US, because of HIPAA, they actually have a legal right to this data. So they can ask the clinic and say, give us the PGTA data that you've generated on our embryos. They can send Herasight this PGTA data and give us some cheek saliva samples or whatever as well. And then we can put this through our wet lab computational pipeline and produce highly accurate embryo genome reconstruction, which we can then use as state-of-the-art genetic predictors of disease risk and IQ. So based on this routine tests in IVF, we can now deliver predictions of IQ, disease risks across like 17 plus diseases, and also we can even genotype rare variants in the embryo thanks to an innovation developed by Jeremy, another guy at Herasight, where you're using, this is probably getting too technical for many people, but we're using these Oxford nanopore long reads to do a more fancy kind of parental phasing. But yeah.
Yeah. So, I mean, in terms of long reads, short reads, just to be clear to the listener, when you do genome sequencing, you shatter the genome and there's like, X number of base pairs. So you have 3 billion base pairs in one haploid genome. And, with the normal, Illumina short read technology, you have like, 200 to 400 base pairs, something like that, maybe 500. Obviously, and then
I think for the PGTA data, it's only 100 base pairs in a single end as well.
Okay,
so it's like really bad quality data.
Yeah. It's really bad quality. So, with nanopore, they're really, really long reads. And when you have a really long read, you can kind of like lay it down and computationally assemble the genome a lot easier, right? And so there's these like synthetic methods where you're kind of using, this as a scaffold for that, where you have the data and you're kind of like leveraging it all together.
Yeah. So Jeremy developed this quite innovative pipeline based on combining different methods of phasing from short reads and long reads with some dynamic programming algorithm that he developed. And then we use that as an input to the hidden Markov model that, I developed in collaboration with Tobias and Jeremy. And putting that all together, it's actually quite remarkable. I didn't think this would be possible when they approached me. I was kind of intrigued by the problem, but I thought that's just such crappy data on the embryos. There's no way this is going to work. But I thought it's worth trying, and we tried it, and we got it to work shockingly well, really. And that if we can get decent quality data on the parents, we can get embryo genotypes that correlate like 0.95 or so with the true genotypes. And really, we're only losing that. So we developed this theoretical result and checked it in simulations and whatever as well. Just using this routine, kind of crappy routine data from IVF clinics, we're able to do embryo selection with efficacy like 90 to 95% as good as if we had the true like Oracle embryo genome, which generally you don't have anyway unless you do deep, expensive whole genome sequencing on the embryo, which is not at all widely available.
Well, I mean, so I know also Joe Pickrell is an advisor, formerly of GenCo and also of New York Genome Center. So there are other academics who are kind of in the Herasight adjacent universe. Yeah, you're looking at the rain out there. Like, we're recording this live, and it is raining in Austin, Texas, and Alex does live in Los Angeles, so I think he's quite disturbed by the H2O coming out of the sky.
I'm not used to seeing this much rain.
Yeah, let's keep it real here, It's like break the fourth wall. But so I mean, as you It's been a while now since you exposed your affiliation. I know you did lose a job opportunity potentially, but what have you heard from academics as they've seen what Herasight has been able to do?
Well, some responded pretty hysterically, I have to admit, and just seemed to they wanted to attack me and the company, and yeah, it seems to be the more sort of ideological academics who, for whatever reason, they kind of, I don't know, this technology sort of offends their egalitarian sensibilities or something, or they just don't like it for whatever reason. I don't know. You have to ask them why they're so hysterical about it. But some reacted very hysterically to it and attacked me in public and whatever. And, I don't really care. Like, I've been dealing with stage 4 cancer, so I'm not particularly afraid of a few academics getting upset. But, yeah, it's sad to see, actually. It's sad to see how negative. There's definitely been an orchestrated effort to kind of try and create a sort of fake consensus or narrow orthodoxy about this technology, like not working, which is like nonsense, or somehow being unethical, which I also think is nonsense. But, you can have a debate about that. But people don't want to have a debate about it. They just want to enforce an orthodoxy about it. And I think they're kind of worried because I'm, somewhat prominent and well known in the field, and it's a lot harder for them to discredit me than it is to discredit some whatever random company that doesn't have anyone very well known working for it. So I think that's part of it. But yeah, there's also been I also, in a way, it was like less negative than I expected, though. I knew there was going to be blowback, and I think there's maybe a recognition now that this is inevitable. It's going to happen. If you try and restrict access to it, all it's going to do is make it inaccessible to poor people, because the rich people are going to find a way to do it. There's a lot of demand for this, I can tell you that. So trying to restrict access to it, it's going to fail. I mean, it's already over, in my opinion. The opponents of this technology have already lost. We've done IQ selection. It's happened. It's over. Like, there's not much point in trying to stop it now.
It's like a fait accompli.
Yeah.
What are you complaining about? It already happened.
Right. I mean, people have I am very in favor of people having their right to, criticize or disagree or whatever. I'd like it if they did it in a way that was, if they actually engaged with the scientific research and the arguments, instead of just calling us or calling me or calling the company names or whatever. But I think that's what they have to resort to, because there aren't really any very good scientific arguments against it, to be honest. And I think that some of the Herasight research it's changing minds. Maybe people are, and in private, a lot of people have said, "Oh, this is really cool," or like really nice work. Like in public, people are still afraid to talk about it. But I think, it'll be a different landscape in a few years. I think it'll be. I mean, IVF was very controversial when it first came around, and that was more from the sort of I mean, there was a lot of, actually, when we first launched it, there was a lot of Christian or like religious people attacking it as well, because of, I mean, that makes sense to me from their moral point of view. So it does sort of raise the ire of both religious conservatives and sort of, I don't know, left-wing or woke academics. So that's one of these weird horseshoe things where they kind of like come together in opposition of this. But yeah, I think that the academic kind of position to be really, really negative and extreme about this technology it's at odds with public opinion. There's actually quite broad public support for doing this for diseases, and there's actually decent level of public support for doing this for intelligence, even high levels of support for doing it in places like Singapore. There's maybe a bit more sensitivity in the West about it. But I think there's probably going to be some kind of a preference cascade about this, that a lot of elite people want to do this for themselves, and once enough people start doing it, then people will forget that they ever opposed it or tried to destroy my career over it, and they'll deny that they ever did that.
Well, I mean, so, we talked a little bit, as we close out, I want to loop back to family-based studies. I mean, I know that's, that is one of the major things you've been focusing on. Like, where are we going with that in the future? Is it just like increase in scale, new methods? Like, what's going on?
Yeah, I mean, I think that...
Well, I do have to reiterate, because you said this really quickly in passing. Siblings, full siblings, their expected value of relatedness is obviously going to be about 50%, 0.5. But genomically, in terms of because of Mendelian segregation and and all that stuff and recombinant, it's going to be in the range of 40 to 60%. And so you could use that to inform the correlation between genes and traits, right?
Yeah, so I've pioneered this study design called family GWAS or family-based GWAS, which uses random variation in genetic variation in families between siblings and from parents to offspring to estimate genetic effects in a way that isn't confounded with various factors like the family environment, the broad population structure, whatever. And this, I think, is a very promising study design for answering some of the trickier questions in human genetics. So estimating heritability properly, estimating pleiotropy or like the shared underlying genetic effects on traits, the shared underlying biology. One interesting application, the most rapid uptake of this work has been from the ancient DNA community. So David Reich he's using this in his using the results of these studies, including my own, to look at natural selection over the past 10,000 years in Europe, because you need to have these estimates of genetic effects that are free from population structure confounding, because that can give a spurious signal of selection. People have been, it's one of these things like everyone wants to find selection in humans. It's like a sexy topic that's been around for like decades now. But it's actually quite easy to go wrong. And people have been burned many times in the past with like declaring that they've found selection on height in Europe or whatever, and then it turns out to be some artifact. So now, David's group has a nice paper looking at this, and they're using some of the results of these family-based studies to try and more robustly assess evidence for selection, and they're finding some nice results on that. Other ancient DNA people are interested in this stuff as well. Then also for answering these questions we were talking about before about, genetic versus cultural transmission, the causes of intergenerational inequalities, correlations between relatives, we need family data for that. So I think family data is important for answering questions where we really need to separate out in a principled way genetic causation from other factors that contribute to kind of associations between genotype and phenotype, or between or that make relatives more similar. So I think that it will become a more important study design going forward, in part because as we move to greater and greater data abundance, we're also going to have greater abundance of family data, which has not been the focus of the way the biobanks have been designed in the past. And just in the end, like you want to have an unbiased estimate of the causal effect, right? Like that's what we want. It's like if you can do a randomized controlled trial, it's better than doing an observational study for all sorts of reasons. And like that's kind of what we're trying to do with family-based studies. It's like the randomized controlled trial version of GWAS compared to standard GWAS, which is more like an observational study.
Yeah, I will say, you have all these like great methods, that you've used your brain to figure out. But I think the key thing is, the brute force reality that we are going to have all the data that we need to answer these questions. Like that is just inevitable.
Yeah,
like, more and more people will be sequenced. We'll get more and more phenotype data. It's inevitable that eventually we will be able to look and understand some of these things, right?
Yeah, we're gonna enter data abundance. I mean, I think it's a shame in a way. There is a lot more data out there than is being fruitfully used, in my opinion. There's an issue in human genetics in particular. I don't know about other fields, but the way it's so complicated and bureaucratic to access data, and people are always trying to figure out ways to make it more onerous. And add in supposed kind of like safeguards that don't really like achieve anything. Like they would never pass a cost benefit calculation if they were done rationally. And if you look at, so UK Biobank is kind of the outlier. That's been the world's leading biobank since it was sort of the first big one as well. And they allowed a much more open access policy than subsequent biobanks have, where they're not so strict about policing what studies you do or hypotheses you investigate. They allow both commercial and academic organizations to access the data. And the impact of the UK Biobank study has been just vastly larger than equivalent studies in the US, which have had much more onerous data access policies. I think, all of us is making up some of the gap now. That's a really nice study from the NIH. But I would, just close, maybe if we're closing, with a call to make it easier for people to access data. There's kind of a sort of monopolistic and ideological practice in human genetics where certain people kind of gatekeep access to data sets, and they gatekeep access partly on like ideological grounds. And there's even been that in all of us, like you have to say, oh, I'm not going to use this to study like group differences or whatever that really means scientifically. I don't know, because like, how do you define a group and whatever. But
I do have to say it's a little funny that population geneticists are asked not to study variation between groups. Just saying.
Right. I mean, it's sort of incoherent, really, if you think about it carefully. Like,
What do you think population genetics is? It's like, if there isn't variation, why are you studying a population?
Yeah, I mean, to me, the within family stuff, what you're doing there is like truly randomized and kind of like causal in some sense. But then once you're not within a family, it's all comparisons between groups in a sense. I mean, GWAS is doing comparison between groups. It's like the group that has the A allele and the group that has the T allele.
Yeah. No, we're in an interesting place. I think we're in a better place arguably than we were 5 years ago during the peak of the late unpleasantness. But we still have a ways to go. I think it's a great time, I think, to do this sort of research, and it's great to have you on the show and get update on what you're doing. Like, we've been talking on and off like for almost 10 years now, on the podcast for like, more than 5. And, I think you've made a lot of advances, obviously, in the science. The science has advanced a lot. That's great. And, good luck to, wherever you're going next, Alex. I really appreciate you coming on and talking about the latest work in population genomics, which I know that this is a topic that a lot of my listeners actually are interested in. But, to be entirely frank, like, not everyone can speak about these sorts of things to lay people, coherently. So it's not always easy to get, the right people, on this podcast to talk about this sort of thing, right? So, I mean, for example, Alex will find this funny. Can you imagine me having Alkes Price on to try to explain any of this?
Well, Alkes talks so fast that it's pretty hard to follow him, even if you know what he's talking about.
That's what I'm saying.
Yeah.
Oh, you go to one of this guy, insiders will know what I'm talking about. You go to his like presentations, and I'm like, okay, like, he is obviously tenured and he is so smart that he doesn't have to worry about all of the rules of how many equations you're supposed to put on a slide.
Well, I think Alkes finished his undergraduate degree at like 14 or something ridiculous like that, didn't he? So
yeah.
His brain works at a faster rate to most people's.
A little bit of the touch of von Neumann, maybe. But anyway, I just wanted to like throw that out there. Well, it was nice having you on, bro. We will talk sometime soon, and I will talk to the rest of you guys next week.
Yeah, thanks, Razib. Always a pleasure to come on.
This podcast for kids. This